Endometrium, Wikipedia audio. The endometrium is the inner epithelial layer, along with its mucous membrane, of the mammalian uterus. It has a basal layer and a functional layer. The functional layer thickens and then is sloughed during the menstrual cycle or estrous cycle. During pregnancy, the glands and blood vessels in the endometrium further increase in size and number. Vascular spaces fuse and become interconnected, forming the placenta, which supplies oxygen and nutrition to the embryo and fetus. The presence of commensal bacteria in the uterus and endometrium has been identified. The endometrium consists of a single layer of columnar epithelium plus the stroma on which it rests. The stroma is a layer of connective tissue that varies in thickness according to hormonal influences. Simple tubular uterine glands reach from the endometrial surface through to the base of the stroma, which also carries a rich blood supply of spiral arteries. In a woman of reproductive age, two layers of endometrium can be distinguished. These two layers occur only in endometrium lining the cavity of the uterus, not in the lining of the uterine tubes. In the absence of progesterone, the arteries supplying blood to the functional layer constrict, so that cells in that layer become ischemic and die, leading to menstruation. Structure. It is possible to identify the phase of the menstrual cycle by reference to either the ovarian cycle or the uterine cycle by observing histological differences at each phase, for example in the ovarian cycle. The endometrium is the innermost glandular layer and functions as a lining for the uterus, preventing adhesions between the opposed walls of the myometrium, thereby maintaining the patency of the uterine cavity. During the menstrual cycle or estrous cycle, the endometrium grows to a thick, blood vessel-rich, glandular tissue layer. This represents an optimal environment for the implantation of a blastocyst upon its arrival in the uterus. The endometrium is central, echogenic, and has an average thickness of 6.7 mm. The functional layer is adjacent to the uterine cavity. This layer is built up after the end of menstruation during the first part of the previous menstrual cycle. Proliferation is induced by estrogen, and later changes in this layer are engendered by progesterone from the corpus luteum. It is adapted to provide an optimum environment for the implantation and growth of the embryo. This layer is completely shed during menstruation. The basal layer, adjacent to the myometrium and below the functional layer, is not shed at any time during the menstrual cycle, and from it the functional layer develops. During pregnancy, the glands and blood vessels in the endometrium further increase in size and number. Vascular spaces fuse and become interconnected, forming the placenta, which supplies oxygen and nutrition to the embryo and fetus. The endometrial lining undergoes cyclic regeneration. Humans and great apes display the menstrual cycle, whereas most other mammals are subject to an estrous cycle. In both cases, the endometrium initially proliferates under the influence of estrogen. However, once ovulation occurs, the ovary will produce much larger amounts of progesterone. This changes the proliferative pattern of the endometrium to a secretory lining. Eventually, the secretory lining provides a hospitable environment for one or more blastocysts. Upon fertilization, the egg may implant into the uterine wall and provide feedback to the body with human chorionic gonadotropin. HCG provides continued feedback throughout pregnancy by maintaining the corpus luteum, which will continue its role of releasing progesterone and estrogen. The endometrial lining is either reabsorbed or shed. In the latter case, The process of shedding involves the breaking down of the lining, the tearing of small connective blood vessels, and the loss of the tissue and blood that had constituted it through the vagina. The entire process occurs over a period of several days. Menstruation may be accompanied by a series of uterine contractions. These help expel the menstrual endometrium. In case of implantation, however, the endometrial lining is neither absorbed nor shed. Instead, it remains as decidua. The decidua becomes part of the placenta. It provides support and protection for the gestation. If there is inadequate stimulation of the lining, due to lack of hormones, the endometrium remains thin and inactive. In humans, this will result in amenorrhea, or the absence of a menstrual period. After menopause, the lining is often described as being atrophic. In contrast, endometrium that is chronically exposed to estrogens, but not to progesterone, may become hyperplastic. Long-term use of oral contraceptives with highly potent progestins can also induce endometrial atrophy. Adenomyosis is the growth of the endometrium into the muscle layer of the uterus. Endometriosis is the growth of endometrial tissue outside the uterus. Endometrial hyperplasia. Endometrial cancer is the most common cancer of the human female genital tract. Asherman's syndrome, also known as intrauterine adhesions, occurs when the basal layer of the endometrium is damaged by instrumentation or infection, resulting in endometrial sclerosis and adhesion formation partially or completely obliterating the uterine cavity. In humans, the cycle of building and shedding the endometrial lining lasts an average of 28 days. The endometrium develops at different rates in different mammals. Various factors including the seasons, climate, and stress can affect its development. The endometrium itself produces certain hormones at different stages of the cycle, and this affects other parts of the reproductive system. About 20,000 protein-coding genes are expressed in human cells, and some 70% of these genes are expressed in the normal endometrium. Just over 100 of these genes are more specifically expressed in the endometrium, with only a handful genes being highly endometrium-specific. The corresponding specific proteins are expressed in the glandular and stromal cells of the endometrial mucosa. The expression of many of these proteins vary depending on the menstrual cycle. For example, the progesterone A receptor and thyrotrophin-releasing hormone both expressed in the proliferative phase, and PAEP expressed in the secretory phase. Other proteins such as the HOX11 protein that is required for female fertility, is expressed in endometrial stroma cells throughout the menstrual cycle. Certain specific proteins such as the estrogen receptor are also expressed in other types of female tissue types, such as the cervix, fallopian tubes, ovaries, and breast. Function: chorionic tissue can result in marked endometrial changes, known as an Arias-Stella reaction, that have an appearance similar to cancer. Historically, this change was diagnosed as endometrial cancer, and it is important only insofar as it should not be misdiagnosed as cancer. Thin endometrium may be defined as an endometrial thickness of less than 8 mm. It usually occurs after menopause. Treatments that can improve endometrial thickness include vitamin E, L-arginine, and sildenafil citrate. Gene expression profiling using cDNA microarray can be used for the diagnosis of endometrial disorders. The European Menopause and Andropause Society released guidelines with detailed information to assess the endometrium. An endometrial thickness of less than 7 mm decreases the pregnancy rate in vitro fertilization by an odds ratio of approximately 0.4 compared to an EMT of over 7 mm. However, such low thickness rarely occurs, and any routine use of this parameter is regarded as not justified. Observation of the endometrium by transvaginal ultrasonography is used when administering fertility medication, such as in vitro fertilization. At the time of embryo transfer, it is favorable to have an endometrium of a thickness of between 7 and 14 mm with a triple line configuration, which means that the endometrium contains a hyperechoic line in the middle surrounded by two more hypoechoic lines. A triple-line endometrium reflects the separation of the stratum basalis and functionalis layers, and is also observed in the periovulatory period secondary to rising estradiol levels, and disappears after ovulation. The endometrium was once thought to be sterile. Instead, a resident, non-pathogenic, and diverse population of microorganisms are present in healthy tissue. The uterus and its tissues are not sterile. The first studies of flora colonizing the human endometrium were published in 2015, were on special populations, and were too small from which to generalize. In one study of 58 women undergoing hysterectomy, investigators looked for 12 species of bacteria. Lactobacillus and Prevotella were the most commonly detected species, and overall density was much lower than vaginal colonization. In another study of 22 infertile women undergoing fertility treatments, Lactobacillus and Flavobacterium were most prevalent, and characteristics of the flora were roughly similar in women who became pregnant and those who didn't. Cycle. Gene and protein expression. The initial stages of human embryogenesis. Clinical significance. Embryo transfer. Microbiome. Vertical section of mucous membrane of human uterus. Endometrioid adenocarcinoma from biopsy. H&E stain. Micrograph of the endometrium. Micrograph of decidualized endometrium due to exogenous progesterone. H&E stain. Micrograph of decidualized endometrium due to exogenous progesterone. H&E stain. Micrograph showing endometrial stromal condensation, a finding seen in menses.